Hello, thank you so much for inviting me to be here today and for presenting at the ISCBacademy this work. We are already performing in the lab for quite some time on the utilization of third generation long-read transcriptome sequencing to study transcriptome complexity. Okay, so you probably know that long read sequencing, the technologies that are developed by PacBio and Nanopore have gained a lot of interest in the last years. And actually, long read sequencing was claimed as method of the year by Nature Method in 2022. And my lab, we have been working for quite some time in applying these technologies to the analysis of the transcriptome. And why are long read methods important of interesting for transcriptome analysis? As you know, in higher eukaryotes, genes are have the potential to express more than one transcript, more than one isoform by the combination of their exon structures. And this results in a number of alternative isoforms being expressed in a particular tissue in a particular moment from the same gene. And probably you know that short read technologies, because the read length is short, it kind of expands the length of the entire transcripts, and it's quite hard to reconstruct this complexity just using the short reads. And in principle, long reads are able to capture that without problems because they are able to sequence full-length transcript in one read, and then you don't need an assembly step to resolve the isoforms, and therefore they should be in principle more suited to for the analysis of alternative isoform expression than the short reads. And why is this interesting? Why is it important to study alternative isoforms? There are many examples in which different isoforms from the same gene are shown to have different types of functionality. And that's because the alternative isoform may have changes in the inclusion of functional elements. For example, an alternative isoform can maybe change an active site, or can also change the way it's binding, for example, to DNA in the case of the transcription factor, or it's binding to other proteins and creating protein-protein interactions. Also, isoforms may vary in the signals that target them to particular localizations in the cell. So by changing their localization, they can also have different functions. And this is not only happening at the coding region of the transcript, but also at the UTRs. You may have differences by alternative isoforms modifying the way in which these transcripts are regulated by cis factors of trans factors. So in general, there is a potential, diversity of functionality associated to different isoforms. So it's important to be able to resolve these structures accurately from the sequencing data. But probably even more importantly is that these technologies have revealed the existence of many novel transcripts, even in very well annotated organisms. And by many, I'm talking about tens of thousands of novel transcripts. For example, this paper on the GTEx collection using long-read sequencing with Nanopore indicated the identification of 70,000 novel transcripts in human tissues. While this other work in Arabidopsis, which is also, a model organism and has been annotated for quite some time already, using PacBio sequencing, the authors were able to identify 170,000 novel transcripts. So it's like these technologies are revealing diversity and a complexity of the transcriptome that we had not seen before. Of course, the question is that true? Is that reliable? I want to show you some how this long read data looks like. Here you have uh, long reads information obtained for this gene, GAPDH, in one particular cell line. And this sequencing data has been gathered by a number of different technologies and then processed and put together in transcript models. And what you can see here is a genome browser view of this gene. Here you have the gene code annotation for this gene that indicates 6 alternative isoforms. And then all this in red is indicating, the diversity of transcript models that were found by a compendium of long read methods. And you can see, this is even cut, because I could not even fit the image in one slide. So here you can see, yes, great diversity, but you can see, for example, that in some cases we have intron retention events, like here. There are some areas of the transcript that seems to be, in general, well resolved, while other regions seem to be more noisy. They have many alternative processing events of these internal exons. So, of course, the questions that we need to ask is how much of this is actual biology, or is maybe the consequence of something else that is not the biology that we are trying to study. So this creates a number of challenges in long-read RNA sequencing. One is to understand how accurate are these calls of transcript models using the long reads. Today, we have already quite a number of tools that are available to reconstruct transcript models from the long reads. And as I mentioned before, they are every single new experiment is reporting thousands of novel transcripts in all species. So how accurate is this? But then once we understand that, of course, we want to be able to use this data to understand more about function and the regulatory role of the diversity of isoforms that we can detect. So questions are like, how do we assign function to isoforms, and whether we are using the same methods to analyze the long reads data that we were using before for the short reads. So my lab has been working on these questions already for quite some time, and we have actually developed tools that are kind of focusing on all these different aspects, and I will be mentioning all of them in this talk with different intensities. One method that has become like a very widely used by the community is Scanty, which is a tool for the quality control of transcript models obtained by long read sequencing. And I will show you how we have used this to do a comprehensive benchmark of the technologies. But then once you have curated the data, of course, you want to, as I mentioned before, say something about the function. So we have this tool, IsoAnnote, to predict or to assign functional labels to isoforms, so with isoform resolution, and TAPAS will then use this information to make questions about regulatory biology involving alternative isoform usage. So for the first part, the quality control of the benchmarking, I'd like to present this project, the Longer GASP initiative, that is like a comprehensive effort to benchmark long reads sequencing methods. So the questions was, so which are the methods, both wet lab and algorithmics, that are providing the best predictions for transcript models using long reads. So in this benchmarking effort, we had different species. We use human, mouse, and the manatee. The idea of including the manatee was to have also like a non-model organism species, a species for which not a lot of genomics resources were available. So we included that in the assessment. We also had different sample types, different cell lines, mixture of cell lines. We also had spikings and the SIRFs, which are synthetic RNAs that are put in the sample, so we know which ones we are adding to our samples, and then we can evaluate with them the different methods. But we also generated computationally synthetic data with different algorithms that are able to do so. Then we also, so that was the samples. We also had different library protocols. The regular one would be using cDNA. So you create cDNA for your transcript, and then it's the cDNA with your sequence. This is available for PacBio Nanopore. But we also tested direct RNA, which is only available for Nanopore, in which you don't generate the cDNA, but you sequence directly the RNA molecule. R2C2 is a library preparation method in which there is, let's say, like a circularization of the transcript molecule, and this is then amplified by PCR and then put into a nanopore. This creates like a transcript structure that can be sequenced like in several passes because of the amplification of the PCR, and that increases the accuracy of the read, of the final read. And then we have a cap trap in which the transcripts are captured from the 5' end. So hopefully that will help to increase, the accuracy on detecting transcription starting sites. And then with these protocols, we have these three sequencing platforms, PacBio, Sequel2 at the time, MinION, and we also generated Illumina data with HiTSeq. So before I continue with this, because it was a large effort, I want to acknowledge the team that was involved in this. The consortium of the team that puts the benchmark together were composed by people who were developing tools or using long-read sequencing, but also people who were able to assess the accuracy of these methods. Important to mention here is Frank, who was a PhD student in my lab and did most of the analysis I'm going to show you now. But for example, very important was also Gloria Segman, who was involved in experimental validation in the project. Kim Fei was involved in analyzing accuracy in quantification. Adam, Jane, and Toby are members of GeneCode and participated also in the curation of the data. And Mark Dickens was really instrumental in helping with the logistics and with the management of the data and the submissions. And this was a challenge. So this was a kind of community competition. That means that the data was generated and then was put in the public domain. And then we asked developers of tools creating transcript models from this data to make predictions on the data. And so these are the names of the labs who participated in the challenge, and that's finally made the benchmarking possible. So so this is the acknowledgement of this work. So we'll be talking about challenge one. There were 3 challenges. One was about transcript isoform detection with a high quality genome. And then we also have challenge 2 for quantification accuracy, and challenge 3 for identification without high quality genome. But I will be talking today about challenge 1 only. There we have several samples, human and mouse. We also have our SIRFs and some mixtures of cells, and then we also have the synthetic data. So just to have a look at the data, I'm showing here different aspects. In this case, on the left, you have read length, and you have 6 different combinations of library preparation and sequencing platform. These 6 combinations were the ones that we generated. So here we have cDNA with PacBio. We also did PacBio with CapTrap. And then the 4 library preparations with nanopore, so the R2C2 and the direct RNA seq, direct RNA with nanopore. So if you have a look at the length distribution of the reads, you can see that there are quite some differences. For example, with PacBio and cDNA, we have the longest reads. Also, reads were long with the nanopore platform, including using the R2C2 library prep. But for example, the direct RNA-seq was shorter reads, and we also had troubles to get long reads or longer reads with CapTrap and cDNA and nanopore. So this is one aspect. Other aspects are represented here. For example, this is then the sequencing depth, the number of reads that we were able to generate, let's say, with equivalent effort. So here is indicated in millions. So you can see with nanopore, cDNA, and CapTrap, we were able to obtain between 10 and 20 million reads, while the other methods were significantly lower in the throughput. And we had around 200,000 reads in each case for the different samples. However, other important aspects is the quality, which is here assessed as the read identity. And then you can see also differences. For example, with cDNA PacBio, we have the highest identity of accuracy of the reads. Also, CapTrap PacBio was similar, and the R2C2 with nanopore had better data quality in terms of identity that the other nanopore platforms. So you can see already that there are quite some important differences in the quality of the starting data with, in general, PacBio having longer reads and more accurate, but the nanopore platform producing more data. And I will come back to that later. So just to give you an idea of what was the magnitude of the challenge, we have these 6 combinations of library preparation and sequencing platform. We have 3 samples. We allow 2 analysis options only using long reads of a combination that's long and short, and that's created 36 data analysis possibilities. Finally, we had submissions by 11 different analysis tools participating in the challenge, and they have different, slightly different properties. Tools like Bamboo, Flames, Flare, and ISOQuant are tools that use the reference annotation to guide the transcript predictions. Some others, like ISOIB, ISOTools, Mandalorian, Spectra, and Talon, they are using the reference annotation as guidance, but not as heavily as before, as the other set of tools. And then Lyric is a tool that is not using the annotation at all. In total, we had 47 pipelines to evaluate. A pipeline is the combination of library preparation, sequencing platform, and analysis tools. So that means that not every tool analyzed the 36 data analysis possibilities. The only thing that we ask is that at least 3 different samples were analyzed with the same set of parameters to see whether results were reproducible. But some tools did not analyze all the data. It was really a lot. In total, 47 combinations. So just like general things that we observed looking across all the transcript models that were predicted are represented here. This is some distributions regarding the sequencing platform, indicating which is the length, the number of exons, and the counts per million. So actually, the 3 samples that were evaluated gave always consistent results. So I will only show one of the samples. But here what you can see, for example, is that the length. So the length of the transcript models and those transcript models that were detected only by the PacBio platform were in general higher, longer than those detected only by nanopore, and those that were detected by both have like intermediate lengths. And the same happens with the number of exons. So only PacBio gave in general like transcript with more exon numbers. And regarding the counts per million, so the expression levels, what we found that in general, PacBio was able to identify transcripts with lower counts than nanopore, even if we had more data in the nanopore platforms. And if we look at the library preparations, we also see some differences. Working with cDNA and R2C2 here. Yes, these cDNA and R2C2 give us also the longest transcript models with more exons and detected with fewer reads than the other two library preparation methods. So again, it seems that general, like working with PacBio and cDNA library preparations or with R2C2 is giving, better performance in these parameters. Okay, so these are general. Then we evaluated the pipelines and the methods, the bioinformatics methods used in the Scanty. This is a tool developed in my lab, which for the quality control. And basically, Scanty was able to do is to analyze the data and also integrate orthogonal data to assess accuracy. So instrumental for scanting is the definition of structural categories that is a way to define how the long-read transcript models relate to the reference. So we have full splice matches, and we will talk about a lot about these terms. Full splice matches are long-read transcript models that match a reference transcript at all splice junctions, like here. They may vary at the ends, but the junctions are matched. We name incomplete splice matches to those transcript models in which they match the reference at all splice junctions except for some of them either at the 5' end of the 3' end of the transcript. So in principle, they look like fragments, although they could also be, for example, alternative transcription start sites. We say there are noveling catalogs to those transcripts that are novel combinations of existing donor and acceptor sites. For example, here we will have, this is, like an novel exon skipping, if you wish. So this splice sites is in principle novel, but is using this donor and acceptor sites that are in the reference. So this is a noveling catalog. And the novel not in catalog is when we have a new donor or acceptor sites that create a completely new splice sites. And then we have some other things like genic introns, what are in the introns, or antisense, or intergenic. So, but we will probably talk mostly about these categories because they are the most abundant. And as I mentioned before, Scanty also is able to lay orthogonal data to support different elements of the transcript models. So we can use short reads to evaluate junctions. We can use cage data to evaluate TSS or quant-seq peaks that are give an indication of the transcription termination site. So we can put this information together and then ask how well the supporting data is contributing to the transcript models. And we generate it in the longer GASP, cage, and quant-seq and Illumina data for these samples. Okay, so I'm going to show you a couple of plots that are a little bit complex, but I hope that I can walk you through interpretation of these. So here we are showing the percentage of full splice matches, which are the known, the good, let's say, transcripts, the percentage of full splice matches versus the percentage of incompletes are these kind of fragments, but otherwise okay. And then also novel in catalog and novel not in catalog transcripts, right? And what we have here is the name of the tools are here represented by the abbreviations, and then in different colors we have the different combinations of library preparation and sequencing platform. Okay, so what can we see here? So what we can see if we look at this circle here is that we see that the tools cluster together regardless the data, right? So it's like, for example, all bamboo samples are very similar in giving a high number of full splice matches, like 90% full splice matches, very few incomplete splice matches, and also very few here of the novel categories, right? And the same holds for ISOQuant, for example, of flame. So we have a number of tools that actually are producing transcript models that really resemble the reference, and that are those that are heavily based on the reference. So in a way that makes sense. Then we have some other tools, like for example, Talon here, Mandalorian. You can see some of the lyric that are kind of giving like an intermediate number, around 50-60% of full space matches. They are able to capture more incompletes. And also here you see the Talon, the Mandalorian, they have a higher percentage of novel in catalog. So they are able to detect more novelty. And these are the tools that, in a way, they have this more room for novel detections. And I am indicating here the lyric, which is not using the reference at all. For example, lyric was the tool that was able to find the highest number of novel not in catalog. Okay. But again, what is interesting here is that it's more related to the tool than actually to the data, how these numbers look like. So the algorithm has a huge impact in the composition of the detected transcriptome. And in general, tools that provide a high number of full splice matches are strongly basing their predictions on the genome annotation. Okay, so this is comparing to the annotation, but what about the supporting data like CAGE and QuantSeq? Remember that we also had that. So here I'm representing how well the 5' is supported by the reference annotations, a little bit like we had in the previous plot, versus how well this is supported by the orthogonal data on the same sample, both for 5' and 3'. And what we see is that, again, so these tools that were kind of using the reference a lot, they are very well supported by the reference. Again, Bamboo here, and ISOQuant, FLIR, they are kind of even given a high support by the reference. And however, it's not as good as the support by the orthogonal data. So you can see that it ranges between 60 and 80% here. And especially the 3' ends, you see this effect that is very well matching the reference, but is not as well supported by the experimental data of the supporting the 3' end in these cases. And on the contrary, those tools that are kind of allowing more novelty are less supported by the reference, so they are not always matching the reference annotation. However, in general, tend to have higher support by the orthogonal data, so they are better capturing the actual, experimental data, especially again at the 3' end, and lyric is somewhere also here in this set. So the tools that use the reference annotations are actually better supported by the annotation than by experimental data, indicating probably some kind of correction of bias towards the reference. Okay, so this is a kind of general overview. Another thing that we evaluated was, what is the agreement between the different tools in methods, in pipelines, in the detection of transcript models. So what you have here is the number and structural category distribution of the transcripts that were, so these are transcripts of unique junction chains, that's kind of the same, that were supported by an increasing number of pipelines. Remember, we have 47 pipelines in total, in which we combine everything together. So this gives a general overview of what is going on here. So what you can see here is that, so this is detected only by one pipeline, and this is detected by all the 47 pipelines. And here you have this in logarithmic scale. What you can see is that the most abundant class is those transcripts that were detected only by one pipeline. And then it decreases as you increase the number of pipelines detecting. And actually detected by all 47 pipelines, there were really few. And if we look at the structural categories, what we see is that these transcripts that were rarely detected by one or few pipelines were enriched in the novel categories. So the blue is the full splice matches that are the known transcripts, and the reds are novel. So And only in those cases that were really detected by many is where we really have an increase in full splice patches. So pipelines agree on the known transcripts, but novel transcripts tend to be pipeline specific. If we kind of try to define a consistent set of transcripts by those that were detected at least by 3 different samples and of 3 different library preparations and analysis tools, and then the distribution is as follows. So it's a great majority are the known, the full space matches, but we also have a significant number of the novel in catalog. So this seems that most of the agreement in the novelty is on the novels. The novel in catalogs are those that have are alternative combination of existing splice sites, which kind of makes sense. But you know, the known is where you have the agreement, and the tools that are more supporting this view are represented here: are Bamboo, Flames, Flare, and ISOQuant, a little bit Mandalorian had, you know, were more in this type of results. Okay, so the previous is just giving a general overview using a scanty to describe, but actually there is not really a ground truth there. You have the support by the experimental data like the cage and the quantic, but we don't really have like a proper ground truth. This is something that we assess with the SIRFs and the synthetic data, and also with the curated data. So here you have the results. Sorry, so here you have the results for the spikings for the SIRFs that were a set of 69 transcripts, and they were different isoforms of the same genes that were mimicking that. And here we have values of sensitivity and precision for the different tools, and for the 6 different there was this freestyle method, but the different combinations of library preparation and sequencing platform. But you can see that in general, most tools did great, both at sensitivity and precision for all the methods. So they are reaching, you know, above 70, 80%, almost 100% in some cases. Like Flare here, for example, of ISOQuant, Bamboo was also very good precision. Lyric was not as great, was working better with PacBio, but not as well with Nanopore. And then Talon also had like a kind of more poor results. But for the others, it's like in general they are good. However, the SIRFs were spikings that were known to the submitters, so they can use the reference annotation to guide these results. So so we are now assessing here the ability to detect novel transcripts. For that, we used the simulating data, in which we simulated both known transcripts, so coming from the reference, but also we generated a number of novel transcripts. And then we evaluated here how well the tools were detecting on these artificial data. So in these plots, you have a sensitivity and precision for the known transcripts, and also for the novel, and this is the inverse of redundancy, that basically different isoforms supporting the same reference transcript, of different predicted isoforms supporting the same reference transcript. And then we did that for PacBio in red and nanopore simulation in blue. I will focus on the PacBio because probably it's easier to explain. So what we see here is that in general, again, we have good values for most tools regarding the known transcript with high sensitivity and precision. You can see them all here. However, when we evaluate the novel transcript detections, then results start not to be as good. For example, Bamboo going from almost 100 precision and sensitivity, we go into values around 50%. Flare, that's a little bit better, especially in combination with short-read data. Mandalorian probably was the tool that has the best performance overall for everything, both in terms of known and novel transcript. But some others, for example, ISOQuant was good at precision, but failed to detect many of the novel transcripts. And the opposite happened with the spectra that detected most of them, but with a lot of false positives and low precision. So this gives an idea that detecting novel transcript is challenging, and but there is also kind of limitation here, is because the library preparation simulates a sequencing error, but probably not as well the library preparation errors. That this is something that we know that also plays a role. So to be able to give an additional layer of validation, we use curation by gene code expert annotators who took a set of 50 loci and look at the reads, and from the reads alone, they annotated these 50 loci with a total of around 300 different transcripts. So we evaluated the tools against this transcript set. So again, here you have the different tools, and these are the different library preparations and sequencing platforms. And precision is in orange, sensitivity in green. And what we found in a little bit, in agreement with the previous result, is that in general for the known transcript we have good precision, and there are some tools that have also relatively good sensitivity, like Bamboo, ISOQuant here, Mandalorian in some cases. Some others not. Lyric was not good at sensitivity, was some missing transcripts and also flames. But in general, there was, relatively good performance at the non-transcript level. But when we are looking at the novel, is when we found that the great majority, if not all, of the tools were really poor at being able to detect these novel transcripts with enough sensitivity. So they were missing most of them. Precision was good in some cases, but that was because maybe there was one or two detected transcripts that turned out to be correct, but there were many of the transcripts that were predicted by gene code curators that were not properly identified by the tools, right? So it seems that we have here a challenge to accurately identify novel transcripts that are not in the reference annotation. So finally, we said, okay, so let's try to validate experimentally some of these known and novel transcripts, especially, right? So can we amplify them? So what we did was to define specific primers for specific isoforms, and then try to amplify a variety of them and see whether we were able to, and then we sequence them again, so we were able to find them back, right? So this was our control just on the gene code curated data. So we were able to validate most of the gene code annotations that were known, also most of those novels, and those that were rejected by gene code in general, we did not amplify them. There was a couple of cases, but this was positive amplification of a mapping error. So actually the transcript model was correct. It was just the annotation that was not correct. So in general, that was our positive control, saying that we were able to validate things that were called by gene code. And then when we look into the data, the results were really, really interesting, because we validated most of the full splash matches of the known transcripts. It was only one that we missed, but it was like a very good validation rate. But also for those novel in catalog, novel not in catalog that were consistently detected by many pipelines, we validate them all. What was really surprising was when we tried to validate those novel in catalog transcripts, even novel not in catalogs, that were detected in very few pipelines, like one or two, maximum three. So what we found is that for the novel in catalog, we also validated most of them. And for the novel not in catalog, it was right around 50%, right? Which is a kind of tricky number because it's like half of them were okay, half of them were not to be validated, although the numbers were not very high. So that gave us a little bit of indication that indeed there are many of these novel transcripts that are in the samples and we can identify them. But to identify that with agreements, it seems to be complicated in some cases. And we also look so how many samples we need to add to so I mean, how many samples were detected those transcripts that were validated, and we can see that even we have a high validation rate of those transcripts that were identified just in one sample. So indicating that there is, a fraction of rare isoforms that are found very rarely in few samples, of 1 or 2 cases, but still they are there, right? So that was kind of interesting of the experimental validation. So concluding this part, I think that yes, the low reads can help you identify transcripts with great precision. But actually, the definition of the transcript models depend more on the algorithm than on the long reads data, because of how its algorithm curates the results. We would say that globally, cDNA PacBio and Nanopore with R2C2 gave the best quality data and probably results. There were quite a number of tools that were okay. As I mentioned, precision is better than sensitivity, especially for the novel, which are transcripts that are challenging to identify. And then we were surprised that many of these novels were actually identified, even if they were detected in a very few pipelines and a very few cases. Okay, so that's a little bit the conclusion of this benchmark. But yes, so once you kind of have a procedure in which you are happy with your transcript models using the long reads, of course, the questions that we also wanted to ask is, what does it mean in terms of functionality and biology? And here I will be presenting some of the results of the lab using isoanode and tapas. So isoanode is kind of a simple idea. So basically what we do here is just to try to annotate using function prediction algorithms, or simply mapping from database using the positions of the annotation of the transcript models on the reference. What are different functional sites, and then we do that at the RNA level with things like, for example, polynylation size, open reading frames, whether they are cis-regulatory elements at the UTRs. We also annotate the microRNA binding sites and also the binding sites of RNA binding proteins. And at the protein level, we do also the typical thing of for example, PFAM domains, transmembrane regions, disorder regions, post-translational modifications, nuclear localization signals, again, using function prediction methods or simply mapping from the databases. And then we do that for every single isoform, also those that are novelly detected by the long reads methods. And this is all put in a snake-make pipeline in which many different sources of databases and algorithms and put together to finally create our annotation. This annotation file, then we can input in Tapas, which is a tool for kind of exploratory analysis and differential isoform usage analysis and functional interpretation of the data, which is kind of a user-friendly tool. We have the possibility to view the transcript structures with the annotations, both at the coding sequence for the different protein domains, but also particular sites, or even the annotation of, for example, microRNA binding sites at the UTRs. So all these display here, and then of course we have like the more systems biology questions. we have used this software with a number of data sets, showing here the results using this simple system in which we differentiated mouse neural progenitor cells into oligodendrocytes and motor neurons, and with different stages, and I will show results mostly of the transition between progenitor cells and oligodendrocytes. So this is one of the results of one of the information that TAPAS can provide, that is what's called that the functional diversity. So basically it's asking the questions, okay, so is the annotation load of the different isoforms of the same gene actually different, so that we can make questions about that? And here, each of these bars represent the number of genes having multiple isoforms that have been annotated in a particular database. For example, here, in which we, for example, this one in PFAM domains, right? So we were able to annotate like 70% of the genes. And then in the darker color, it's indicating how many of these genes had transcripts of isoforms with different annotations in this database. And that was around, like 70% of these 70%, so a majority. And you can see that this is the case. There might be not always annotation, for example, and signal peptides, we only have a few genes with that kind of information, but also most of them have isoforms with different annotations. So it makes sense to ask these kind of questions. TAPAS has like a traditional analysis tools, like, for differential expression analysis. Basically, what we take are the gene models that we have obtained with the long reads. These isoform resolve functional annotation obtained by TAPAS, and then we also have, gene of transcript expression matrix, in which we have the expression levels. This can be quantified with long reads or with the short reads, but we have some quantification values. And then we do this kind of like the functional diversity analysis that I mentioned before, and this differential analysis as well. For example, the things that you can do is to ask whether you have changes in the splicing, and also what is the magnitude of the change in isoform usage, right? Or whether you have a switching event. And the magnitude of this change in isoform expression of isoform usage is what we call total isoform usage change. And this is, for example, one of the plots that we can show in which we put together, which is the changes in the differential expression, the fold change, like for the whole gene, versus the change in the usage of the isoform usage. So that gives us an indication of how genes are regulated by differential gene expression of differential isoform usage. And then you can see that there are many genes that, for example, here that have big changes in expression level, but not at the isoform. But there are some other cases, for example, this gene here that has actually no change at all at the protein at the gene level, however, has a huge changes in the utilization of alternative isoforms, right? So that you can see how genes are differentially regulated. Once you have this information, you can ask, and which are the functions that are represented by this gene? And here, this is also one of the plots that TAPAS gives you, in which you are seeing how different functions are differentially regulated at the level of gene expression in orange, or at the level of differential isoform usage in this differentiation process. And then you can see that most processes are regulated by both, and most processes are mostly regulated by change in gene expression. But in some cases, for example, brain development of importing to cells, and I think we have here also myelin sheath. So these are biological processes that are mostly regulated by changes and isoform level, right? So that you really need to capture that to understand how they are regulated. And then we have this TAPAS-specific analysis that we developed specifically to understand better changes and functional content when you have different isoform changes, in which basically we take each functional element and then we evaluate the differential inclusion across isoforms as a function of the condition. And we can do the same also for UTRs, and we can do that for polymyelination signals and so forth. If we check which are the functional domains, the functional labels that are mostly regulated by differential isoform usage, we see that disorder regions, PFAM domains, but for example, also microRNA binding sites are frequently regulated, differentially included when you have a different isoform expressed. And also what we can see is that if we evaluate how many, what is the amount of functional content that we have as we transition from progenitor cells to oligodendrocytes, we see that in oligodendrocytes, so in the mature cell, we have in general a higher number of inclusion of functional elements that in the progenitor cell. So meaning that isoforms that are expressed in the differentiated cells have more functional load, functional domains for almost all databases. This is also an example of the things that you can find. This is one of the genes that were differentially, that had differential isoform usage, but not differential gene expression. It has 4 isoforms, and you can see that 2 of them are high in progenitor cells and low in oligodendrocytes, and 2 others have the opposite pattern. When we look at the functional content, so this is the TAPAS annotation, we see here a difference between these 2 patterns that is associated to the inclusion of a nuclear localization signal. And what happens is that the isoforms that are expressed in oligodendrocytes here, they have the inclusion of the nuclear localization signal, but not in the ones that are expressed in the progenitor cells, where the cytosolic fraction is kind of similar. So basically, the change in isoform is controlling the localization of the protein at the nuclear fraction between and this is changing between progenitor cells or oligodendrocytes. And this is a function that, this is a protein that is involved in the catenin signaling pathway. And we can verify these kind of predictions, we can verify it in the lab. And finally, some other things that we can also, do with TAPAS is the analysis of the length of the UTR. For example, here we are comparing motor neurons and oligodendrocytes. We see that in general, motor neurons have longer UTRs. This is associated to the inclusion of a number of sites. These are the different stages, and you can see how, for example, many microRNA binding sites are especially included as the differentiation progresses, and these are microRNAs involved in axon regeneration, and you can see them here that because of how the UTR is modified, you have inclusion of particular microRNA binding sites that are present in the motor neurons and not in the oligodendrocytes. So with that, I come to my final conclusions. So trying to wrap up everything that I mentioned today here, I think that, so definition of isoform models from long reads is possible, but it's still challenging, especially for novel transcripts. We are working hard in creating the tools to curate these models, but also try to provide some functional insights and which is the possible alternative function of isoforms in terms of the functional domains and what is the consequences of alternative splicing. And as I mentioned before, we are seeing a large number of novel isoforms that are systematically found in single samples and low expression levels. So maybe we should, think how do we do with that? Do we annotate them all, or we are just call them, sample specific? So the question is whether every single RNA molecule deserves to be called an isoform of how do we approach this analysis of this diverse pool of rare but existing RNAs. And with that, I want to finish my presentation with acknowledging the people in the lab. I previously acknowledged the Longer Gas team but this has been mostly work of people who already left, Lorena de la Fuente, and Manuel Tardáguila, and also Ángeles, and yeah, so thank you for your attention. I'm happy to take questions.
Excellent. That was very informative. A lot of data packed in there. So we do already have some questions in the Q&A. The first one here, you note that simulated data does not simulate library preparation errors. How impactful do you suspect those library preparation errors may be on performance, or can that not be estimated as it's instant dependent?
So very, very. I think the library preparation method has a huge impact. Actually, we have seen this. I have not presented the results here, but you can probably see this in the paper. We observe that quite a lot. With the different in the longer gaps with the different library preparation methods that we tested. So remember, we had 4 different ones, and then what we can see there are differences in the ability to capture either the 3' or the 5'. There is a contribution of the degradation of the RNA, so that if you have degraded RNA, you will have a lot of incomplete displaced matches that are just degradation product, and it's kind of tricky to differentiate that. I think at present, the most, one way to go is to use orthogonal data. For example, if you use cage data, you can resolve some of these um, problems in detecting transcription start sites, which is kind of one of the major issues today. But yeah, I think that is more a question of the library preparation than of the sequencing platform.
Gotcha. Okay, that makes sense. Our next question, is there any explanation for ONT shorter reads compared to PacBio in cDNA experiments?
I don't have one. That's what we got. I think this is already some time ago. So probably now this is not that much of an issue. But yeah, that's what we got with the nanopore. I don't know why that was the case, to be honest.
Fair enough. One of those little mysteries. Um, could you explain how experimental validation is performed?
Yes. Okay. So what we did was to look through the transcript models and try to identify primers that will amplify specific isoforms. That was very, very challenging because there are many isoforms, and it may not be always easy to find a region that is specific for one. So, but that's what we tried, and we look at the isoforms that were expressed in the tissue, but also those that were annotated, and then we will amplify, like not the whole transcript, because maybe we are not able to amplify a very long transcript, but we will amplify a region that we can confidently say that is specific for that isoform. And then these amplicons were sequenced. And if we recover the expected sequence, we say that this isoform has been validated.
Following up on that, just a personal question. If you got a sequence that you didn't expect, say you got those isoforms that did match and so it was validated, but you also got extra data, would that diminish the validation, or would that be just another question that you'd have to look into?
I think that's another question that we had to look into. We did not really got into that, but that was my fear, actually.
Mhm.
But this is not what we found. What we found was that we were basically amplifying what we were expecting to amplify, or we were not amplifying a lot at all. Understand? So I would say that 90% of the cases, the amplicons were what we expected.
Wow, very nice.
But sometimes we did not amplify, right? So that's the
mhm.
thing that we do not validate. Yeah. Yeah. Sorry. Go
Still,
ahead.
that's That's a really nice confirmation number. 95% is always nice. Another question. If you only had one pipeline, would you say there is a way to select novel transcripts with higher probability of being experimentally validated?
That's a great question. Um, I think if your transcript models have all the features of a good transcript model, probably yes, because this is actually what the manual curators did. They sometimes believed transcript models with just one reads of a few reads. But if all display functions were, either annotated or were supported by short reads data or recon data, that's what they used. They would say, okay, this looks like this is a real isoform, and then if there was also patterns of conservation, if there were also good support by orthogonal data at the TSS and TDS. So if everything looks like it's okay, even if it's a novel isoform detected with one pipeline. I would say that it's fine, because again, what we are seeing not only in these experiments, that we find a lot of transcripts or RNA. I call them more RNA molecules than transcripts, because I don't know what qualifies an RNA molecule to be upgraded to a transcript. But we find these RNA molecules that are just expressed in single samples. Yeah.
Interesting. In the same vein for that, is there a specific like a transcription pipeline that you prefer working with? Like you'd mentioned PacBio and Nanopore, and was there one that just it was nicer, it was easier for whatever reason you liked more than the other ones?
Okay, so that's a tricky question, because so in this longer gas project, I was the jury. Okay, I was the evaluator. So I did not compete with my own pipeline, the ones that we are using
Ah,
in the lab, and that's of course the one I prefer, right?
makes sense.
So basically, I mean, there are things that are great, but again, so you have to be cautious when trying to call new transcripts. But if you are trying to recapitulate the reference, Flare, Mambu, Mandalorian, and ISOC1 will be great. What we do in our lab is to use the cDNA PacBio approach, but then use the scanty, what we call the scanty filter, so the scanty curation tool, in which we evaluate a lot of these, features of good quality of transcripts to discard or to retain those, problematic transcript models. And that's what we normally do. So if you are able to have some orthogonal data, even if it's not of the same sample, but is in the same organism, it really helps to correct things. And yeah, so that's what we do. This pipeline is this like the Scanty3 pipeline, and you can find it. It's now, the paper is in the review, but you can find the bioRxiv paper describing how we do things. But yeah, I could not participate with that in the competition.